to today's journal review session. The topic that I brought today is aging-associated changes in transcriptional elongation influence longevity. So this was published in the Nature paper this year, April 12th, so not long ago. The key point I really want to make by reviewing this paper is that the transcriptomics is an important study for aging. That's the key point. And the down below is the figure for transcriptional elongation for those who you don't know. This is the enzyme RNA polymerase II that is involved in the transcription of DNA to RNA. And this over here is the string of DNA which is wrapped around the protein called histone. The more densely packed the histones are, so the DNAs, they get less transcription, and the more accessible the less dense regions are which that gets transcription. And as you can see in the blue over here, this is a nascent RNA which becomes the active molecule and become a protein at last. And the elongation by the RNA polymerase II is actually governed by the speed of the enzyme. The things that this paper wanted to convey us is that there's a tendency that faster speed of RNA polymerase II elongation with aging, and reduction of its speed extends lifespan. This is the first result. First, they explain how they actually measured the polymerase II speed. So polymerase II speed can be measured with total RNA-seq. They use total RNA-seq, specifically the intron region of it. So over here you can see this is one of the gene region of Drosophila, the frazzled gene, and its first intron. And as you can see over here, the slope represents the speed, and the lower it gets apparently in this experiment, the shallower the slope, the faster the elongation. Once they set up how to measure the speed, they actually experimented across the entire model organisms, including the human. And what they could achieve is that the x-axis is the, as you can see, the elongation speed or the speed of RNA polymerase II, and y-axis is the different experiment they did. And the older, as you can see over here, the older individuals, old versus young, senescent versus proliferating, that's for the cell. The older it is, the faster the tendency of the elongation. And as you can see over here, this is the interventions, anti-aging interventions such as insulin IGF signaling inhibition and dietary restriction. As you can see, they get slower transcriptional elongation. To validate their observation, they've actually did the experiments on the C. elegans and Drosophila, and when they actually introduced this genetic variance that is located in the homologous, this is the homologous genes for RNA polymerase II, they could see that they get the extended lifespan when the genetic variant is reducing the RNA polymerase II speed. And that's conserved for both C. elegans and Drosophila. Next, what the authors did was to actually survey the epigenetic mechanism that is underlined by this aging-associated polymerase II speed. And what first they did was to actually naively look at the histone levels. And the x-axis is represented by different human cell types. And when they had the proliferating young cells, there is high expression of histone or H3. And for senescent older cells, they have decreased expression of histones. Excuse me. And as you can see over here, the hallmark of aging is actually the loss of histones. And if we increase the level of histones, that means, and theoretically, we could decrease the aging speed or increase the longevity. And experimentally, they've actually shown this by overexpressing the histone, which is represented by H3-GFP and H4-GFP. They could show the slower elongation rate, and this actually correlated with the lower level of nuclear P21. And what P21 is also a hallmark of aging in senescence. The entry of P21 in the nucleus happens in the older cells. And as you can see, as we overexpress the H3 and H4 histone proteins, we have decreased, compared to the control with only GFP, there's a decreased level of nuclear P1221. And as for the underlying biology of this phenomenon, the faster polymerase II speed is actually bad for transcriptional fidelity, or otherwise known as just bad transcription or wrong transcription. And this low transcriptional fidelity have been recurrently reported for its relatedness to the aging. And these are the plots that we've already seen for elongation speed, but now the x-axis is circular RNA expression and erroneous splicing and consequent mismatches rather than elongation rate. And as you can see, this is a kind of similar plot. The older the individuals are, the more circular RNA it's produced, and the older the individuals are, the higher the mismatch levels, and vice versa for other genetic variants reducing elongation speed and dietary restriction or anti-aging intervention. So erroneous splicing, accelerated transcription, erroneous splicing events, advanced age and shortened lifespan, circular RNA formation, accelerated transcription, the elevated formation of circular RNAs, and age-associated phenotypes as a result. And finally, when the authors actually did an enrichment analysis to see the function of the genes that were associating polymerase II speed, they've actually shown the enriched function of the metabolic pathways, including the catabolic process, lipid metabolism, organic cyclic compounds, and so on. This is a conclusion. Dysregulation of transcriptional elongation reduces cellular and organismal fitness and may therefore contribute to aging and related disease phenotypes. And as for the application, our dataset actually lacks total RNA-seq samples, so it would be difficult to proceed the same experiment through, although we can try this in mRNA-seq or the just small number of total RNA sequencing that we have. But on the bright side, changes in splicing and circular RNA formation across the age can be surveyed with our dataset, although the resolution will not be great. So there is hope that we can actually do this kind of experiment in our lab. And lastly, I want to finish off by introducing you to the lab called the Center for RNA-Mediated Healthy Longevity, RNA Aging for short, located in KAIST. And this is actually run by PI Lee Sung Jae. And this figure was brought up to our lab before, and they've kind of did a similar kind of study that the splicing is affected by aging, and this event is actually useful for measuring the biological age. So I want to, in the next session of journal review, I want to review this paper.